Hey, Mr. P here. In this video, we're going to talk about the structure and function of the digestive system. So we're going to talk about all the anatomical structures that belong to the digestive system, talk about what their roles are within the digestive system, and then we'll pick up with kind of the structure and function of both digestion and absorption and assimilation in future videos. But for now, let's kind of kick off this this foundational video in where a lot of the structure and function is discussed. So, some key terms that we need to kind of identify up front before we get into kind of the nuts and bolts of digestion is obviously looking at how digestion is mediated, meaning how does digestion actually happen? A couple key processes that play into the overall process of digestion is ingestion first. We obviously have to eat food. Okay, ingestion is the act of consuming food. We obviously do this through our mouth. We then take the ingested material and we, using a series of chemical reactions, actually take the ingested food and convert it into smaller and smaller molecular forms. We use enzymes and often a series of enzymes to actually facilitate this chemical process, where we're actually taking larger compounds or polymers and we're trimming them down to individual monomers a lot of the time. We then have to absorb those monomers into tissue cells. A lot of times we use the nearby blood supply, or we use the circulatory system in order to actually send the molecular forms or those monomers, that nutrition, to all of the tissue cells. And then finally, transport, using the circulatory system, delivers the small molecular nutrients to our body cells, and that's where the assimilation and the true use of those molecules happens. Okay, so digestion is an enzyme-facilitated chemical process. It requires ingestion first, digestion second, absorption third, and transport fourth. Okay, that's how we get food from outside of our body into our body, digested, absorbed, and transported to all tissue cells in our body. Okay, that's how we actually nourish the body cells that make up us as a multicellular organism. Okay, individual cells that make up individual tissues cannot consume food on their own. It requires the digestion and absorption and transport of those nutrition, okay, or of that nutrition to individual body cells throughout the body. Okay, so really, without kind of going into any more detail about those processes, we need to really dig into what the digestive system is, what it entails, what the structures are, and what the processes and roles of those particular structures are. And so I'm going to start at the mouth because the digestive system is actually a one-way flow, or a one-way tube. Okay, it's a series of interconnected tubes that work in one direction from the mouth all the way to the anus. And so we obviously start this discussion or dive into where digestion occurs or begins, and that is the mouth. So the mouth is where obviously chewing happens. It's actually the place where the first enzymes are utilized, okay, in the form of salivary amylase. More of that will come in a little bit. Saliva glands are, or the salivary glands are actually associated with the mouth. Okay, they are the glands that secrete saliva, which obviously is going to contain salivary amylase, which is the first enzyme I kind of just talked about. Food will be ingested through the mouth. It'll be mixed with saliva. It will then transport down a tube known as the esophagus, ultimately emptying the contents into the stomach, where obviously the pH is greatly reduced. The pH of the stomach is around 2, which is much lower than the pH of the mouth, which is slightly alkaline at about 7.8. The stomach pH will play an important role in digestion, which I'm going to talk about in a little bit. Okay, from the stomach, we move past a series of kind of accessory organs. One of them is the liver, another one of them is the gallbladder, and then the third is the pancreas. Pancreas is kind of this little hidden greenish organ, which is incredibly important when we start talking about pancreatic enzymes and the addition of the enzymes to the digestive system. After the pancreas is the opening of the really long coiled up tube, muscular tube, known as the small intestine. And the small intestine is broken up into kind of two parts. The duodenum is the upper end of the small intestine, and the ileum is the lower end of the small intestine. Both of them belong to the small intestine. Okay, the small intestine is this entire structure, but this entire structure is broken into two halves. The duodenum is the upper half, and the ileum is the lower half, and they both have very specific roles, which we'll talk about, like I said, in a minute. Post small intestine, we come to the large intestine, which is actually a much larger tube than the small intestine, with an incredibly different role than the small intestine has. Once we exit the large intestine, we get to the rectum, and then ultimately to the very end in the anus. And so you will see that this is a one-way flow. It's not a cycle, right? It doesn't cycle or circle back on itself. It is a one-way flow by which we ingest food and ends with the egestion of feces. Okay, so what are the roles of some of these structures? The mouth is obviously where we started this in-depth look at the organs as we worked from top to bottom. But as we work through this digestive system, I want to break it down into kind of zones. And we're going to talk about what happens at each of these zones kind of as a standalone process so that we can more thoroughly understand the importance of each process within the whole system, which is the digestive system. So in the mouth, we have teeth, we have saliva, we have salivary glands, and then we have obviously salivary amylase, which is the first enzyme our food will actually be subjected to. Chewing is called mechanical digestion, okay? And mechanical digestion is the part of the digestion that includes a mechanism in the teeth that is actually grinding the food into smaller bits. That is mechanical digestion. That is not chemical digestion. Chemical digestion includes or employs the enzymes that we're going to talk about in a minute. Mechanical digestion is still a form of digestion because it is actively breaking the food from larger components into smaller components, but is not actively breaking down polymers to monomers. It is just breaking down food size into more manageable, smaller sizes so that one, it can actually be swallowed, and two, it increases the surface area and decreases the volume of the food items so that the enzymes are more efficient at working towards the hydrolysis that needs to take place. In the mouth, the saliva really has two roles. One, it's going to soften the food and make it easier to chew the food, but it's also going to allow the food particles and the food substance that you're chewing to stick together and produce a structure called a bolus. A bolus is a food particle or is a group of food items that is all swallowed in one chunk, right? And so we obviously need to lubricate the esophagus. We need to make the bolus easier to transport down the esophagus, and saliva moistens the food and makes it easier to swallow the bolus. Salivary amylase is the actual gland that produces the enzymes and the saliva and will secrete the enzymes via the saliva in order for chemical digestion to start. Amylase, as we will discuss later, is actually a really, really important enzyme when we talk about chemical digestion of starch. Amylase actually breaks starch down into individual maltose molecules, which hopefully sounds familiar to you. Maltose is a disaccharide which is composed of two individual glucose molecules bonded together. Okay, once we leave the mouth, we enter the esophagus and we employ a process called peristalsis. Peristalsis is a muscular contraction that moves in a wave-like motion to move the bolus into the stomach. Okay, so peristalsis is moving a bolus using longitudinal and circular smooth muscle tissue. And we're going to talk a lot more about what peristalsis is in a little bit, but we basically at this point need to know that it moves by contracting the smooth and longitudinal muscle slightly behind the bolus on its way to pushing the bolus into the stomach. Okay. The stomach is the next stop for the food, and so muscular contractions continue the mechanical digestion within the stomach. Stomach acid actually reduces the pH, and the pH of the stomach is around 2, which greatly reduces the pH of the food at this point, because the pH of the mouth is typically around 7.5, and that reducing pH actually serves two purposes. One, it kills any bacteria that might be present in or on the food to help keep you safe and safeguard you against foodborne pathogens. But also, it denatures the salivary amylase so that it can kind of reset the system and produce enzymes and, as you'll see in a little bit, actually restart the enzymes post-stomach. Okay, and again, we'll make that clear in a minute. Pepsin is a protein digesting enzyme, and it is employed within the stomach to actually begin the digestion of proteins specifically. And the stomach is really only one of only a couple locations where proteins are actually going to be digested. Okay, again, that is in the stomach. Then we move into the small intestines. Again, like I said, the first half or the top half of the small intestines is called the duodenum. This is where the bile that is stored in the gallbladder is secreted via a duct into the duodenum, post-stomach. So we are now past the stomach. We worked into the first part of the duodenum, and this is the location where the bile that is stored in the gallbladder is going to be secreted into the small intestines. That's really important because it neutralizes acid and emulsifies fats. Why do we need to neutralize the acid? Because the contents of the stomach that are leaving the stomach, including the food material, have to obviously be neutralized because, again, the pH of the stomach is a 2. The pH of the small intestines is just above or just slightly higher than 7. If we do not neutralize, or if we fail to neutralize the pH, then the contents that are moving out of the stomach are going to greatly reduce the pH of the small intestines, which will have severe consequences, not only in denaturing the enzymes that are secreted by the pancreas, but also make this environment unfavorable for the living bacteria and the microorganisms that are living symbiotically and mutualistically within the gut. Okay? Pancreas then plays a role in this particular location as well. The pancreas releases a myriad or a cocktail of enzymes, including lipase, pancreatic amylase, and trypsin, as an example, all at the same time through this duct, and will flood the duodenum. And so this upper end of the small intestines is really where a lot of the digestion takes place. That's a really good distinction. Okay? Once the digestion occurs on the upper end of the small intestine, then the contents move to the lower end of the small intestines, which is called the ileum. The ileum constitutes or is made up of the lower half of the small intestines and is where a lot of the absorption takes place. So upper end of the digestion or upper end of the small intestine, the duodenum, is where the digestion takes place. the lower end is where the absorption of those nutrients takes place. You will see in later videos that there are structures called villi that are numerous in population within the lining of the small intestines, which is going to greatly increase surface area and greatly increase the efficiency by which we can absorb nutrients. Once you leave the small intestines, specifically leave the ileum of the small intestines, you will enter the large intestines where digestion and absorption doesn't happen. Okay, there is a strong distinction between the role of the small intestines and the role of the large intestines. Small intestines is the digestion and absorption of nutrients. Large intestines is specifically the reclamation or the recycling and the returning of water to the bloodstream. And so if you can think about, as the contents of the small intestines work through the small intestines, we're absorbing what we can absorb, we're leaving what we can't absorb, and then all of that contents, or all of those contents, flood the large intestines where we try to reabsorb a lot of the water, leaving behind semi-solid feces, which will be stored in the rectum before egestion can happen, which is the feces being forcefully removed due to muscular contraction through the anus as feces. Okay, so again, this is the alimentary canal. It is a one-way canal that is muscular in nature from the mouth to the anus and is really the site of mechanical and chemical digestion through a series of different locations from mouth and ingestion to anus and egestion, or the removal of waste. It is really, really important as you look through the IB syllabus and notice that there is a skill associated with this particular diagram. You could very well be expected to produce out of memory a drawing of this digestive system during your final examinations, and so in order to do that and to make sure that you're prepared, I would encourage you to practice drawing this several times in order to ensure that you receive full marks. You may also, kind of as a caveat to this particular skill, be expected to annotate a particular diagram, which is likely kind of what we did here, right? The diagram was in place. I then put all of the labels and arrows pointing to the proper structures. So, what is this alimentary canal? The alimentary canal is a muscular tube from start to finish. It doesn't matter if we're talking a cross-section of the esophagus, if we're looking at a cross-section of the stomach, if we're looking at a cross-section, in this case, of the small intestine. All three of those locations are going to have the exact same kind of anatomical structures, and it's essentially a lumen area, or an open tube that is surrounded by two layers of smooth muscle. The inside layer is called the circular layer of smooth muscle, and the outside layer is called the longitudinal layer of smooth muscle. What does that mean? That means that these two layers that sit on top of each other actually crisscross in a 90-degree angle and run perpendicular to each other. Okay? It is important to note that the smooth muscle associated with these two layers is controlled by the autonomic nervous system, or the ANS, which is the same division of your nervous system that plays a role in maintaining heartbeat, maintaining respiratory rates, maintaining a lot of the unconscious behavioral thoughts and the instinctual responses and the feedback mechanisms and the homeostatic balance that exists within our body. Okay? You don't have to physically think about moving the bolus down your esophagus. You don't have to think about the churning that happens within the stomach. You don't have to think about the digestion and the absorption of those nutrients as the food contents travel the smooth or the small intestines, right? All of that stuff happens automatically without thought. Okay, and so that is a part of that autonomic nervous system, the ANS. The circular layer can contract, the longitudinal layer can contract. Okay, both layers can also relax, just like any other muscle fiber. They can contract and relax. And so when the circular layer of smooth muscle contracts, this inside layer actually makes a circle. And what I mean by that is, if you look at these two layers, you will see that the fibers on this inside layer are actually running around, lengthwise around the lumen, and are producing the circle, which is why they're referred to as circular layer. The longitudinal layer is actually running lengthwise down the length of the tube. Okay? Again, these are positioned at right angles to the circular muscles, and because the longitudinal layer and the circular layer can all contract and relax at the same time, you get this process called peristalsis, which is kind of the way that the two layers work in concert or in tandem with each other. And that's what allows us to move the bolus from basically mouth to stomach, or from stomach through small intestines, or small intestines to large intestines, without much thought. So what is this process, peristalsis? Peristalsis is the contraction of both longitudinal and circular muscle, smooth muscle fibers. You will notice, just like in that last diagram, that the longitudinal muscle fibers are running lengthwise along the outside of this particular tube. The circular muscle is running, again, you can kind of see that it's speckled because they would actually be running circular. And so we're looking at a cross-section of them. And so for peristalsis to happen, the longitudinal muscle has to expand in front of the bolus this way to open up and reduce pressure so that the bolus can slide towards that decreasing pressure. The contraction of the circular muscles behind the bolus actually pushes in and increases pressure by decreasing the volume of the lumen, and so by simultaneously increasing pressure in this location as well as decreasing pressure out in front of the bolus, it actually causes, due to this muscular contraction, the sliding of that bolus down this tube. Okay? Contraction just behind the food mass keeps the food mass moving through the canal, and so it works in one direction. It doesn't back up. It doesn't move in a non-desirable location or direction. It moves in a desirable direction, and in this case, moving down the esophagus and ultimately emptying into the stomach. Okay? Peristalsis, again, is used in the esophagus. It's also used in the smooth intestines. It's also used in the large intestines, but it also is used by the stomach. The stomach obviously has a lot larger volume than the esophagus and the small intestines, and so the process looks a little different. However, we call specifically the peristalsis associated with the stomach churning, and what that really means is that the stomach actually contracts and relaxes in order to kind of knead the food matter that's in the stomach, in order to fold in the enzymes that are pepsins, right? The protein digesting enzymes that are widely available, okay, or readily available in the stomach. It also kneads in or incorporates or folds in the stomach acid to help kill the microbes that might be coming into our digestive tract through the food. Okay? Now, peristalsis moves in two different speeds. Peristalsis associated with the esophagus is really, really fast, and peristalsis with everything else, especially with the intestines, is very, very slow, and that's by design. Peristalsis associated with the stomach or at least getting materials to the stomach, right, with the esophagus has to be fast, because from an instinctual response, we as consumers are kind of at a disadvantage in our environment by spending time consuming. So we want to kind of move around our environment, get as much food into our stomach, which is kind of like the storage vessel we're containing all of this kind of ingested food matter. And we want to do that relatively quickly. So we want to be able to get food content into our stomach quickly. We also want to be able to store it in our stomach, but we want the back half of this digestive system to work slowly, because if food moves through the digestive tract too fast, meaning if peristalsis happens in the intestines too fast, we're not actually going to be able to absorb all of the nutrients out of the food, because it's just moving through the small intestines too fast. If the food moved through the small intestines as fast as the food moves through the esophagus, we would essentially absorb nothing from the food that we ingested. Thankfully, it slows down to a point where it makes the absorption process pretty efficient, because it allows the food to pass through that tube slowly. Okay, that's where we're going to end this particular video. If you have questions, make sure you bring them to class. Otherwise, I will see you on the next video. See ya!